The overall goal of this procedure is to prepare Drosophila oocytes for the cytological examination of spindle assembly and chromosome orientation during meiosis I.
This method can help answer key questions in the meiosis field, such as how spindle bipolarity is established and how chromosomes become bioriented in the absence of centrosomes. The main advantage of this technique is that the oocyte membranes that block antibody penetration are effectively removed, which allows immunofluorescence.
To begin, pre-coat the inside of a 5 ml tube in Pasteur pipette with PTB to prevent oocytes sticking to the surfaces. Add 100 ml of Rob's buffer to a blender and then add 100 to 300 anesthetized flies and pulse 3 times for 1 second. Filter the resultant slurry into a 250 ml beaker through a large mesh with pores of around 1,500 micrometers. Let the filtrate settle for around 2 minutes, and then aspirate the top layer, removing as many large body parts as possible. Filter the slurry again into a 250 ml beaker through a smaller mesh with a pore size of 300 micrometers. Rinse the first beaker using additional buffer in the coated pipette to collect the remaining oocytes. Let the slurry settle for 3 minutes and then aspirate all but the bottom 10 ml. Pour as much of the 10 ml as possible into the coated 5 ml tube. Let the slurry settle for around 2 minutes and then carefully remove the liquid.